Hi everyone, my name is Astrid Hyldbakk and I'm a PhD student at the Norwegian University of Science and Technology in Trondheim in Norway. My PhD project is mainly based on development and characterization of a new polymeric nanoparticle-based drug delivery platform in close collaboration with the research institute Sintev. Today I will talk about one of our characterization methods for nanomedicines, a high-throughput method for drug release measurements in complex media. Okay, so first just a little bit about Sintev. Sintev is one of the largest independent research institutes in Europe. And for more than 70 years, Sintev has developed solutions and innovations for society and customers worldwide in a broad and multidisciplinary field. Last year, we sold for more than 3 billion Norwegian kroner, equal to 300 million euro. The organization is based on contract research, and none of our owners receive any form of dividend, as Sintev is a not-for-profit organization where all financial surplus is invested in scientific equipment and skills and expertise. And Sintev is heavily involved in nanomedicine technologies, including the whole value chain from drug discovery, development of new methodologies for nanoencapsulation, physicochemical characterization, in vitro characterization, and in vivo characterization in rodents. So my focus today is on drug release measurements as a part of the physicochemical characterization step. So as you all know, drug release is an important parameter to characterize when developing a new nanomedical formulation, and it involves incubation of the formulation in a medium before the released drug is separated from the encapsulated drug and quantitated at specific time points. Typically, drug release measurements are performed in PBS or other salt buffers with a physically relevant pH, but these buffers do not mimic the very complex in vivo environment very well. So it is therefore preferable to study drug release after incubation in a biological medium, such as blood or blood plasma or serum, or other biological fluids such as cerebrospinal fluid. But incubation in such protein-rich media does add complexity to the experiment, as you have to take the interactions between the drug and the proteins into account. So it can therefore be beneficial to use stable isotopes as reference materials to compensate for this challenge. And isotopes are versions of an atom with detectable variations in neutron count. So like you see here for hydrogen. Protium, the standard hydrogen atom, has 1 proton and 1 electron, and this gives a molecular weight of 1 Dalton. Deuterium, on the other hand, is often called heavy hydrogen, and it has 1 proton, 1 electron, and 1 neutron, giving it a molecular weight of 2 Dalton. But regardless of this difference in molecular weight, the hydrogen atoms behave almost identically, and we can call them chemically equivalent. This figure shows the drug doxorubicin, the original version of the drug, and the isotopically labeled version where 3 hydrogens are exchanged with 3 deuterium atoms. These 2 molecules will have the same biological activity in vivo, and the only difference is the 3 Dalton mass difference. So by adding a known concentration of the labeled drug to the sample, we can measure how the labeled drug is affected, and we can use this knowledge to compensate for the interactions between our drug of interest and the proteins in the sample. And we can also quantitate the amount of released drug, including free and protein drugs, protein-bound fractions. And encapsulated drug. The importance of measuring these drug fractions is highlighted by recent FDA and EMA regulatory guidances, recommending their measurements in order to establish generic nanomedicine bioequivalence. This can be done as shown in the figure made by Sarah Scottson and her group when they presented this methodology as a part of the nanotechnology characterization laboratory in 2015. So first you have your nanomedicine with an encapsulated drug, and you add this to the plasma, and you add a known concentration of the drug analog, isotopically labeled one. And then you will have an equilibrium between the released drug and proteins in the sample. And this equilibrium is formed at a comparable fashion for the unlabeled and the labeled drug. And then you filter your sample through a membrane, and then only your free drug will go through the membrane. And you can quantitate the amount of free labeled drug in the filtrate, and this can be used to calculate the protein-bound unencapsulated and encapsulated drug fractions in your sample by these equations. So just a bit about the workflow. This illustration shows the workflow for the analysis. This is done in a well plate format with 1 plate per incubation time point. So at a given time point, the plate is taken out from incubation and you add a stable isotope analog. And then the content of the plate is split into 2 pathways, one for quantification of the total drug amount in the sample and one fraction that you spin filtrate, and you get only the free drug fraction in your sample. Then both these plates are extracted by an organic solvent, and you use the organic phase for drug quantification. And all liquid handling steps are performed by robots, as shown in the pictures and in this movie. The robot lifts the specific plates into position, takes them out of the incubator, and dispenses and transfers sample between wells and mixes the content, and can also lift the plates directly into a plate reader, for instance. This gives a much higher sample throughput, as we don't have to spend time on manual pipetting or sample handling. And additionally, robotic sample handling increases the precision and accuracy, as human errors are limited. And the reproducibility and reliability are also increased. The 96 or 384 well plate format allows us to include many parallels and variables, such as many concentration levels of the sample and different release media. This picture shows a typical incubation plate, where we have incubated our nanoformulation in PBS and human plasma and different cell culture media. But to quantitate the amounts of drug and the isotope labeled version of the drug, mass spectrometry is needed. This is an instrumentation that allows for specific separation and sensitive detection of molecules, such as small molecule drugs, based on their molecular weight. So this allows us to separate molecules with only small differences in molecular weight, such as our drug and the isotopically labeled analog, for example, doxorubicin and doxorubicin D3. So by analyzing their instrumental responses against standard curves, we can quantitate the concentrations of each substance in the sample, and we can calculate the percentage of drug release from the formulation, and then we get curves looking like these. These are some example results from applying this method to a liposomal drug carrier and a drug polymer conjugate. So figure A showing the liposome, shows the calculated drug release after incubation in human plasma for up to 24 hours. And we have included 3 different concentration levels of the sample in this setup. Figure B shows the drug release of a drug polymer conjugate in both PBS and human plasma for up to 1 week. And this graph clearly shows the differences in release kinetics in human plasma versus PBS. And it illustrates the importance of studying drug release in a biologically relevant medium or relevant to the conditions that you work with. So to summarize, the presented method allows for drug release measurements in complex protein-rich media, such as human plasma. And you can also run this method in blood or plasma samples from in vivo pharmacokinetic studies after you have taken blood samples at specific time points after injection. It allows for assessment of released, including free and protein-bound drug and encapsulated drug fractions, robotic liquid handling with high precision, and simultaneous analysis of several drug concentration levels, incubation time points, and release media, for instance. So thank you for listening to this presentation, and I'm happy to discuss the method with you. And please let me know if you are interested in knowing more about the method, our facilities, or if you are interested in a collaboration with us at Sintev. So thank you.
Thank you, Astrid. We have already 1 question in queue. Nadezhda.
Thank you very much. Yes, I have a question. Actually, at first I want to say thank you for the presentation and for the demonstration of such an interesting and powerful platforms, because yes, it could help us to solve a lot of really important problems. But I want to ask you, you showed the data, or the experimental data, and you said about using the plasma. Which kind of plasma is it? You mean some plasma from the donors which are taken in the moment of study, or which kind of?
It depends on your study. Sarah Scottson, that has presented this method or published it, they have both done, like, you incubate your sample in plasma samples, for instance, from blood donors, that you just use it as an incubation medium. But as I said at the end here, you can also use plasma from your experiments where you have injected your animal with a nanoformulations, for instance.
Well, I asked because from the one side, it's really important to use the plasma, for example, with donors, if we have some donor study or animal study. But from the other hand, this point, the using of plasma is a moment which could change the result dramatically, because the plasma components, they're not really stable in the time. Yes, it depends on the meal, it depends on the time of a day, it depends on some genetical even characteristic, and we cannot use this factor. We cannot think about the data precisely in our study. There are, like, like black box. What do you think? Could it be standardized somehow?
Yeah, I totally agree. We have actually discussed this in our group, and we wanted to do a big scale screen of this to study release of standardized nanoformulations, for instance, doxyl or a very characterized one, in different plasma samples. For instance, from a person that just had a really fat-rich meal, or
it could be really important.
I agree, but what we do to try to avoid this is that we pool plasma samples from many or more than 3 donors every time, so that we.
So you try to just enlarge the number of points.
Yeah.
Well, I guess it's best solution in this situation, because there are too many factors to think about. Thank you very much. Well, I will look for the results.
Yeah, thank you for the question.
Ulf has another question. Please, Ulf, can you?
Yeah, very simple, I guess, to answer two questions is, do your center has also expertise? I mean, it's a topological labeling is an interesting methodology to study tumor metabolism or metabolism of cells. Do your center have expertise in metabolic, so to say, flux analysis of cells, particularly in vivo? And the second is how I maybe missed it at the beginning. How would a collaboration ideally work, scientifically, or work you on contract based? So thank you for this.
Your last question, I would say both.
I was what? Muted?
No, I hear you. But for your last question, I would say both. If you're interested in or have any more questions, please just send me an email or
okay.
That would be really nice. But for your first question about metabolomics, our group has not focused on this field deeply. We have some. We're going more and more in that direction, but we are also collaborating with a lot of groups with more expertise in this field. Mm-hmm. But we have instrumentation that allows for. Yeah.
When you talk about metabolomics, Ulf, I know that at Sintev you have, so we have similar instruments also, but at Sintev you have multi-TOF imaging systems. Am I right?
Yep.
Yeah. So we also perform metabolomic profiling by multi-TOF imaging system, which is really nice. Only problem is if you have a drug that has a very low concentration in tissue samples, then you can see changes in metabolomic profile, but you cannot really see drug. This is tricky thing with this MSMS. Yeah. So, but
okay. So you should on this aspect. Okay.
Yeah. So it's I know quite well. So I think you have Bruker machine or
yeah Bruker machines and a lot of different Agilent machines as well. But our multi-TOF is Bruker.
Yeah, multi-TOF imaging is a Bruker. Yeah. And are you able to perform also the small molecules and also proteins, or just because there is a Bruker machine which is devoted to proteins and proteomics?
This is not my specialty.
Okay.
I've mostly been working on small molecular drugs, on quadruple or triple quadruple instruments, and that we have plenty of, and they are our working horses, and they have a really tight plan or tight schedule every day. So, but we also have multi-TOFs and
yeah. So, are there any questions from audience to Astrid? Okay, so because I wanted to ask additional questions regarding this technique. So I'm curious, when you talk about this binding, drug binding, and as far as I understood, you add isotopically labeled drug after extraction from some cell extract, or?
No, we're not using any cells. You just add your nano formulation in media, for instance, human plasma. And
okay, so it's just characterization in biological media.
Yeah.
But is it possible to use such technique if you treat cells and then see what happened?
I would say you could just take out a sample and run it in the same fashion. Then you would see how much is released at a specific time point after you have had it in a at those conditions for.
And did you try perhaps to add the isotopically labeled drug before to nanoformulation before like cell treatment, and then treat cells with such mixture, and then make the analysis like you explained?
That we have not, but it sounds like an interesting way of testing it. Yeah.
Yeah, because it would really help to solve questions about the fate of nanoformulation and drug on it after they reach and they enter the cells.
Yeah, for sure.
Yeah. Okay, thank you. So more questions from audience? Okay, no. So Astrid, thank you very much for this presentation.